Now that we understand how prokaryotes do transcription simplistically, we have to address how do eukaryotic cells make it a little bit more complex. So first of all, binding of the RNA polymerase to the promoter is not good enough for eukaryotes. The RNA polymerase actually can't bind by itself to the promoter, and it needs the help of what are called transcription factors. Transcription factors are these other proteins that will bind to the promoter first, and then will kind of entice the RNA polymerase to come and bind as well. And the binding of the transcription factors and the RNA polymerase to the promoter creates this large complex that's called the transcription initiation complex. And once this complex forms, then transcription can actually start, and the RNA polymerase can chug along the DNA, making its mRNA. As part of this transcription initiation complex is the promoter region of the DNA. So let's talk a little bit more about this place where the RNA polymerase needs to bind. The promoter contains the transcriptional start point, so it tells the RNA polymerase where to start transcribing the gene, and then it usually extends several dozen nucleotides upstream of that start point. Also, promoters are going to contain what is called a Tata box. This is part of the promoter that's critical for binding of the transcription factors so that you can create the transcription initiation complex. Let's take a closer look at the Tata box. So here we're focusing on, again, gene and the double-stranded DNA. We've got the actual coding part of the gene in darker blue, and then the promoter in that seafoam color. You'll notice the promoter here contains the start point for transcription initiation. It also has this region called the Tata box. It's called the Tata box because it's got a lot of T's and A's in it. And so the Tata box is where these extra transcription factor proteins bind to the promoter first, and then they encourage the RNA polymerase to come along and join them at that promoter, along with several other transcription factors. So this whole chunk of stuff, the DNA, RNA polymerase, and the transcription factors form this transcription initiation complex. Once that's formed, then the RNA polymerase can start doing its job. So it'll pry apart the two DNA strands, figure out which one is the template, and then start adding, in red here, the new RNA nucleotides in a 5' to 3' fashion to start building that primary transcript. Lucky for us, elongation is really no different in prokaryotes versus eukaryotes. And so we'll just go into a little more detail here that applies to both kinds of cells. Remember, RNA polymerase is kind of a one-man band, so it can unwind the DNA strands by itself. It'll stabilize the single-stranded DNA by itself. It adds new RNA nucleotides complementary to the template DNA strand by itself, doesn't need a primer or anything like that. The RNA polymerase really only untwists about 10 to 20 bases of DNA at a time. It really only needs to access a small portion of it to make sure it can get to that template DNA strand. I remember the RNA polymerase will build the new mRNA in a 5' to 3' fashion. So just like DNA polymerases, RNA polymerases also add to a free 3' end. And the other cool thing is that you can actually have one gene being transcribed simultaneously by multiple RNA polymerases. I'm going to hop back a couple of slides here just to use this one as an example. So once this RNA polymerase has moved past the promoter and is down here, the promoter is free and available to accept new RNA polymerases. So a second one can hop on here and start chugging along behind the first RNA polymerase, making more and more of these RNA transcripts in a very short period of time. And this can happen both in prokaryotes and eukaryotes. Okay, and then lastly, how do we end transcription in eukaryotes versus prokaryotes? For bacteria or prokaryotes, we kind of addressed this earlier. It's pretty simple. Every gene is going to have a sequence in the DNA that's called the terminator, and the polymerase will run into the terminator. It'll recognize it and say, oh, I'm finished with transcription of this gene. It'll hop off, it'll release the mRNA, and the mRNA is complete. It's fully baked, and it can immediately be used for translation without any further modification. In eukaryotes, it's a little bit different, not nearly as clean as it is in bacterial cells. So I'm going to go a couple slides back just to use this particular figure as an example. So in eukaryotes, down here when we're looking at termination, there is no terminator sequence at the end of the gene. So the RNA polymerase is kind of naive in a way maybe a little stupid, in that it just continues chugging along the DNA even past the end of the gene, and it has no idea that it has actually finished transcribing the gene. And so it continues running through, and at the end of the gene, there's something that's called the polyadenylation signal sequence. It's a lot of adenines all in a row. And the RNA polymerase doesn't recognize it, so it transcribes it and continues moving on. Once that polyadenylation signal sequence kind of juts out here in the mRNA past the RNA polymerase, there are enzymes that are inside the cell that are kind of watching this whole thing happening. They're like, hey guys, look, that RNA polymerase has gone past the polyadenylation signal sequence and he doesn't even know it. He's just continuing to go. And they chuckle a little bit, and then they say, well, okay, we should probably help the guy out. And so these other enzymes will come along and they will cut the RNA free from the back of the RNA polymerase past that polyadenylation signal sequence. And so the RNA then will float off free, and the RNA polymerase, it's still chugging along this whole entire time, and it kind of turns around, looks back, and says, my RNA is gone. I guess I must be done. And so it'll hop off the DNA, release the last little chunk of RNA that it's made, and sort of anticlimactically finish its transcription of that gene. That little extra segment of RNA that doesn't really code for anything that's been created after the polyadenylation signal sequence, it'll get chopped up and the nucleotides can be reused in that way. All right, so in eukaryotes, just to review, RNA polymerase will transcribe past that polyadenylation signal sequence, and then the transcript is released about 10 to 35 nucleotides past that sequence by these other enzymes that are found inside the cell.